So we'll start off with a summary of meiosis cell division, but I thought what would be prudent to do initially is just remind ourselves about what's taking place in mitosis cell division so we can then draw a comparison between the two. Right, so with mitosis, we're going to look at, I'm just going to use humans as an example. Being given extra information, you could be asked about potentially any organism with different diploid numbers of chromosomes. Again, if we remind ourselves about what diploid means, so if we start off with our parent cell, what diploid means is that you have got, it's the total number of chromosomes within a cell where those chromosomes exist as homologous pairs. So if you remember, N represents the number of different chromosomes within a species, and on each of those chromosomes you're going to have different genes coding for different polypeptides at, once again, different loci, different positions along the length of the genes. So N is the number of different chromosomes, and importantly there are two copies of each of that type of gene. One of those chromosomes, two copies of each of those chromosomes rather. You inherit one of those chromosomes from your mother, and so they're referred to as the maternal chromosomes, and you inherit one of those chromosomes from each homologous pair from your father, and therefore they are referred to as the paternal chromosome. So in humans there are 23 different chromosomes and they exist in homologous pairs. So in terms of the actual diploid number, it's 46. We know then that during the end of interphase in cell cycle, the end of interphase, the cell prepares for mitosis cell division, and there are a whole range of different things going on, but one of the things that takes place is semi-conservative replication. So each one of those 46 chromosomes will replicate itself, and so now we end up with 92 chromosomes in total. In mitosis cell division, the aim and the purpose is to produce genetically identical cells in terms of the genes that they contain, and the number of chromosomes will be maintained. So when we have the cell division, we'll end up with the diploid number of chromosomes in each of those daughter cells. And that's mitosis cell division. That's the purpose of it. Let's compare then and contrast with meiosis cell division. As I said, this is just an overview, this is just a summary of the process, and we'll look at the details subsequently. In humans, and I stress in humans because we will look at other organisms, in humans and in all mammals, and generally within, generally he says, within phylum Chordata, within kingdom Animalia, generally, there are exceptions to the rules, generally what happens in meiosis cell division is it is used to produce gametes in these organisms. It is used to produce sex cells. Let's use humans still as an example. So we've got 46 chromosomes within the diploid cell, and exactly the same in meiosis cell division within the cell cycle. And we'll look at the details and the application of meiosis in humans later on. So this whole process is taking place in the testes of males, it's taking place in the ovaries of females, but the detail of that we'll look at later on. So in these diploid cells, in its cell cycle, towards the end of interphase, exactly the same process is going to take place. Semi-conservative replications going to occur, and we'll end up with 92 chromosomes. Now, there are two main functions of meiosis cell division, and we'll look at each of them individually. But as I said, meiosis cell division in humans and most chordata animals is to produce gametes. And importantly, those gametes must have the haploid number of chromosomes. We'll see the importance of that in a moment. And so to produce the haploid number of chromosomes, as you can see on this initial summary diagram, in order to achieve that, one of the key main differences between meiosis and mitosis cell division is to produce haploid gametes. There have to be two cell divisions. So initially in meiosis cell division, we have the first cell division that takes place. So we'll end up with two daughter cells, and they will have 46 chromosomes. We are not going to describe, after the first cell division, these two daughter cells as being diploid. You'll see why when we look at the detail of meiosis a little bit later on. It's a technicality, but it is correct nonetheless. So they have 46 chromosomes, but they can't be described as being diploid. We'll discuss that later on. So in order to achieve the haploid number of chromosomes, each of those two cells must then undergo another cell division. So we have one cell division, and then we have another round of cell division. So we'll have 23 chromosomes in each of the daughter cells. These are all gametes. These could be sperm, these could be ova. Let's say for argument's sake that these are the ova, and this is within a female, within their ovaries. And so we're going to describe these gametes as being haploid because they will contain one copy of each of the different chromosomes present within that species. So that's one of the functions of meiosis cell division. Now the purpose of that, the reason why they must be haploid, these gametes must be haploid. If these are ova, if I draw, obviously not to scale, if we then draw a sperm and it also is haploid, the reason why gametes must be haploid is so that when fertilization takes place and the nuclei from the sperm and the ova fuse together, they will then produce what's referred to as a zygote. We'll look at all of this detail in a completely different topic. But when those two nuclei from the two gametes fuse together, what you can see that happens then is that in the next generation, so this zygote is then going to form the offspring, in the next generation, the diploid number for that particular species is maintained, and that's really important. If you didn't have gametes with the haploid number of chromosomes, they would not end up with the diploid number of chromosomes for that particular species, and in which case this zygote would not go on to form a fetus, an embryo, etc. So function number one of meiosis cell division: produce gametes with the haploid number of chromosomes to allow the diploid number of chromosomes in that species to be maintained from one generation to the next, and so on and so forth. The second function of meiosis cell division, as I'm sure you'll know, is to produce genetic variation in, initially, the gametes formed. So each of these, we'll see in a moment, I'm going to illustrate this. In each of the gametes formed, they will show genetic variation to one another and also compared to the parent cell. And therefore, when fertilization takes place, the offspring eventually will show genetic variation compared to both parents. So to illustrate the role of meiosis cell division in terms of creating genetic variation, I'm going to just look at this diagram over here. Let's look at a particular organism, a particular species where they don't have huge numbers of chromosomes. Let's say that we're going to look at this particular species, and it has a diploid number of 4. So it's going to have a total of 4 chromosomes, and it will have 2 copies of each of the different chromosomes. To just help to illustrate the principle behind genetic variation being generated in the gametes, I'm going to use 2 different colours just to represent, within a homologous pair of chromosomes, the fact that you've got, as I said before, a maternal chromosome, so one of those chromosomes that you've inherited from your mother, and I'll use a different colour to represent a paternal chromosome, which you inherit from your father. Okay, so let's say then, for argument's sake, I'm going to use black is going to represent maternal chromosome, and red is going to represent the paternal chromosome. So you can see we've got one homologous pair of chromosomes here. Again, we discussed before, the only way in which we can initially say that they are a homologous pair of chromosomes is because they are the same length, they're the same size. If we had the ability to have a look at their genes, we'd see that they have the exact same genes in the same locus, in the same position, running along the length of those chromosomes. So there's homologous pair number 1, and here is homologous pair number 2. So diploid number 4, 1, 2, 3, 4, but remember, diploid number is the total number of chromosomes that exist as homologous pairs. So we've got homologous pairs of chromosomes there. So what we will see is that within meiosis cell division, there are 2 main processes that take place which are going to generate variation in terms of the combination of chromosomes, and therefore also the combination of genes within the gametes. We'll look at these processes in detail later. The first process is called independent segregation. Now, sometimes books refer to this process as independent assortment. On our particular syllabus, it's referred to as independent segregation, but independent assortment, it's the exact same thing that's taking place. This, as we'll see a bit later on, this process has the ability to give gametes different combinations of maternal and paternal chromosomes. I'm going to illustrate that in this diagram, but there's a second process that takes place also within meiosis cell division, which is called crossing over. Crossing over has the ability to actually swap and exchange genes between both maternal and paternal chromosomes. But the point is that these 2 processes have this effect of generating genetic variation. It's important that we stress that it's genetic variation within the gametes. I'm not showing the 2 cell divisions. This is just a summary. We're going straight to the 4 gametes. And so what's important is that you will end up with genetically varied gametes. If we just concentrate on independent segregation, just to illustrate the general point of genetic variation, remember we're going to have each of these gametes is going to be haploid. So it must have the haploid number of chromosomes. And if we know that the diploid number is 4, then haploid means that the number of chromosomes is going to be halved. So we know they're going to have 2 chromosomes in each of them. But what's a really important point, and this is a common mistake that students make, is that there has to be, as we've discussed in this little diagram down here, each of the gametes has to contain 1 copy of each different chromosome. And that's really what haploid means, a copy of each different chromosome. So if we look here, the first gamete can contain the maternal chromosome, the longer one from the initial diploid cell. What we couldn't do is then draw in this gamete the longer paternal chromosome. That can't happen. That can only be, there must be 1 copy from each homologous pair. Therefore, this gamete could have this combination of chromosomes, and we can just work through that logically. So the second gamete could have the longer maternal chromosome and then the shorter paternal chromosome. This gamete could therefore have the longer paternal chromosome and shorter maternal, and then finally this gamete could have the longer paternal chromosome and the shorter paternal chromosome. So that fulfills the criteria of being haploid. So it has half the number of chromosomes of diploid, but remember the gametes must contain 1 chromosome from each homologous pair, plus you can see that each of these is different genetically in terms of their makeup. So that's the role of meiosis cell division, to produce haploid gametes, so that when fertilization takes place, the diploid number of chromosomes is maintained from each generation to generation, and then each of those gametes is going to show genetic variation, so that when fertilization takes place once again, the offspring is going to exhibit genetic variation compared to both parents.